Hey guys, can you hear me? Yep, Ryan says yes. Yep, good, okay. Let's wait for a few more people. I was late a few minutes, so. Are we ready for tomorrow? I don't see any positive reaction. Rita says no, no. Okay, that's not the answer. That's incorrect answer. Try again. Why you're not ready? Come on, it will be fine. We won't panic, right? So let's see how many of you I see here. I don't know where is Bri. Vera says too much information for final. Well, sweetie, that's the class. I mean, it is microbial physiology. It's like a biochemistry. I told you at the beginning, I don't know how much you remember first class, but I did say that it will be a lot to remember. And it's way better if you start studying like from the first class. Because if you didn't study like anything, it will be disaster. But I'm pretty sure you guys are fine. I mean, according to interactions that I had in class, I think that people will be pretty fine. Like if you understand, you won't have to worry about it. So Brittany is still not here, which is surprising, but I will have to start. She might join later. So here it says we are recording. So it should be fine. And I hope I will receive recordings afterwards, because last review session, which wasn't really informative, like you had review documents, so it should be fine. I'm hoping I will receive the recording, because I see in left corner of my screen, it says recording. So I should be fine. Now, so what I will do, I will share my screen with the lecture, with the PowerPoint presentation that I made for review session. And later on, you can either ask me questions or type questions in chat. And please be aware that I cannot really tell you what will be the question, and like that wouldn't be ethical, but I can help in guiding you. So let's see how, share screen. Yep, that's it. Can you all see the screen? Can you? Let's see. Yes, so you can see the screen, good. So, okay, welcome to the final session, final review session for the final exam that we have tomorrow, December 19th at 2 p.m. in cab 235. So for those who didn't attend often to classes, it's a Central Academic Building 235, that's a classroom that we used for our lectures. So here is how the exam looks. So to some of you, I wrote it's 25 questions. It's actually 28, 26 and 7, 29, but it is total 100 marks. So, and last 4 are definitions. So you will see now. So all exam is divided in, all exam is divided in 3 sections. So first section will be multiple choice, which has 15 questions, 2 marks each. Second section are short snappers. So there are only 6 questions. Each of them is 5 marks. Short snappers, basically short answers. So I'm not asking you here to write anything in detail. So basically just understanding, basic understanding. So that's what you need to know. And I will lead you through everything. Section 3, the end of the second section is bringing 30 points as well. So for section 3 is probably the hardest one, and there are 2 concept questions. So one of them is very long, that one that has 18 marks, and The other one is 14 marks and a little shorter. The last 4 are definitions, basic definitions, and those 4 are bearing 8 marks total. So, section 1, that multiple choice. Again, as probably you probably know that I really hate multiple choice questions. I know that it's very easy to make mistake over there, but during on my USRI evaluation, I got a comment that I should give some multiple choice questions, and I'm following what students say. So, and when I talked to other professors, they suggested me that I should definitely have some multiple choice questions because there are students that prefer it. So I accepted critique. So there are 15 of them. Each of them is 2 marks. First hint, go through all quizzes. All quizzes are available for review till tomorrow noon. So go through each single question on the quiz, especially those questions that were problematic. Make sure you know very well. So this section has pretty much a question from each unit. Some units I have 2 questions, total is 15, you had 11 units. So just go through those questions and make sure you understand. Now, here are some hints for those multiple choices. NAG and NAM, PBP, protein penicillin binding proteins. NAG and NAM, monomers that are involved in peptidoglycan biosynthesis. So know what they are. Know bonds that they are forming. Bacterial cell division proteins, MIN-E, MIN-D, FTSZ. Chemotaxis, know phosphorylation and methylation. Know what each of those key protein does. What is methylating, what is demethylating protein, which one is first going to be phosphorylated, which one is the other one. Memorized by key A, key Y. A is first letter in the alphabet. Brittany was explaining to you how can you memorize. Know proton motive force components. What is making proton motive force? Delta psi, delta pH. So no chemiosmotic theory, no electrical and no electrical component of the electrical and chemical component of proton motive force. Fatty acid biosynthesis. No enzymes. So Fab enzymes, HGA and I, know what each of them is doing, especially that one that is target for antibiotics. There are Fab-I, final reductase, Fab-H, the one that does condensation. SEC-A and SRP for protein translocation. Know in which system, like SEC, co-translational or post-translational, which one is. Know which type of energy is turning on co and post-translational translocation system. For secretion systems, know landmark for each and really go very carefully through quizzes for each secretion system. Stringent response, very well understand REL-A and spot-E differences. So REL-A mechanism, what is causing REL-A based activation of stringent response? It's stalled ribosome when there is mutation in tRNA. Neutrophilic adaptations to pH changes. So know what do they produce. So we don't talk here about acid, no differences between acidophiles and neutrophil adapted to low or high pH. So remember, if it's neutrophil, PMF is generated through N delta psi and delta pH. If it's not, then it's only one, delta pH or delta psi, depending on is it low or high pH. Oxidant and reductant, know how to define this. That's very important. What is oxidant, what is reductant, and know an example for each of them. Now, as about central carbon metabolism, understand basic differences between EMP and EDD pathway. How do you know that bacteria is running EMP or EDD? Be aware that glyoxal shunt is a version of TCA, but bacteria that use it they use acetate as a carbon source. 4 pathways, EMP, EDD, TCA, glyoxal shunts, and I forgot to write here pentose phosphate pathway. Key enzymes. That's what you need to understand. Iron and manganese reduction. Know which form is oxidized, which form is reduced, because if form is oxidized, like iron 3+, that can be electron acceptor, and if it's Fe2+, it can be donor and it can be oxidized. Now, understand the role of humic acid in metal-based respiration. What humic acid does? Breathing solids, remember, when you have iron or manganese oxide. Understand the eukaryotic denitrifiers, especially what is in plum and what is in trophosome, who resides in trophosomes and what resides in plum. Comamox, exactly know what Comamox is doing. It is also ammonia oxidizer, but it's complete oxidation, not the same as Anamox. Anamox is on denitrification side. So that's all for section 1. For short snappers, section 2, which is 5 marks each. Know very well glutamine synthase regulation with low and high ammonia or high and low alpha ketoglutarate. So know what's happening with the gene, how it's, so in high ammonia, what will be the activity, but you have to know how to basically explain. Again, look quiz questions. Yeah, excuse me. I explained it very well in correct answer, but also look review document because that document contains pretty much summary. Understand microbiome use for health predictions. So in that lecture, we talked about how human diet affects different types of bacteria or microbes in our gut and how that affects our signaling and what can happen later if it's disturbed. We'll know very well Anamox and specific structures that are decorating these Anamox organisms. In terms of metal regulation modes, so we talked about various metals. One of them specifically must be regulated because it's toxic in high concentration, and there are a few modes to regulate that. This bifurcation enzyme hydrogenase. Know what is bifurcation enzyme, where this enzyme donates electrons, and which organisms are able to use this bifurcation enzyme. Microbial fuel cell, know compartment details. What is in oxic, what is in anoxic, what organisms breathe in microbial fuel cell, where electrons go from, where they travel from. Section 3, this one is the hardest. So my expectation is that you will all mess up mostly on this picture. So I had to have one of those questions because somehow I have to separate A+ from B students or C students. So there will be a picture of a cell, which is not in lecture. So it's cell that we created. So when you look, and everything will be written, there will be no structures to recognize. There will be a word of specific metabolites. And when you see those metabolites, you will have to recognize which pathway that is. So what you will search for is what is produced and what is consumed. So, and this is from units 6 to 11, actually 6 to 10. Note to conclude what is carbon source, what is the fuel, and how they assimilate carbon. Then know what is energy source. I pretty much gave you a hint now. It has either it has both. So sometimes carbon is also an energy source. Here you have a carbon source, but there is also another separate energy source. So which organisms can use that? And know whether this is bacteria or archaea, because there are certain differences between bacteria and archaea, and you will be asked that on that picture. The longest question that carries 18 points is only one pathway from lectures. There are different modes of this pathway, and I'm clearly asking you what to respond. So I'm giving you all modes, and I ask, what is this, what is this, what is that? And it clearly says how many marks each of them bears. And last four definitions. It's a basic physiology and bioenergetic. Again, this is a hint. Remember what I was telling you that you must know always. And that will be definitions that I will ask you. And these four definitions will give you 8 points. Now, last thing, there are 2 bonus questions. One from my research, the other one from Brittany's research. You don't have to respond neither of them. But if you respond one of them correctly, but only one, like if you respond 2, no. If you respond only one correctly, you can miss 2 multiple choices or 1 short snapper. So I think it's pretty cool. So that would be all from review session. And now I'm open for all questions if you have. I don't see anything on chat. Does that mean you don't have questions? Vera asks, I wonder if the marks, Vera is asking if the marks of total on E-class is accurate. No, because I still have to manually override that because you all will get 10%. So what I might do, and Brittany was telling me that I should do that, and I will. Just I didn't have time. I will calculate where all of you stand without final, and I will send you. And I will probably, if I have enough time, I will email to each of you separately. I just didn't have time to do that. So there is another question in the chat. It's about the bonus. So if we do the bonus question, we can leave 2 multiple choice. No, you cannot leave it. You have to, so if you do it and you make 2 mistakes, you cannot just leave it blank. You have to try. So Rita is asking, can you please explain carbon labeling principle? Is it relevant for final? Yes, it is relevant for final. And yes, I can explain. So let me just see if there are more questions. No. So let's see. So let me, Rita, give me just a second to wait for a few more questions, and then I will explain carbon labeling principle. Is that okay? Yep. So ask a few more questions, and then we can go through carbon labeling. And if you guys can remember something more, please ask. And of course, I will post this to the e-class. Do we have more questions? Melissa asked, what is the difference between quinone and menaquinone? It's a different type of carrier. It's just different molecular structure. Menaquinone is present in anaerobic bacteria. Quinone is present in aerobic bacteria. It's just if you remember when we talked about salt stress, there is a ubiquinone 8, the one that acts like a glue. So it's also different type of quinone, just a different molecular structure. And Soren asks, do we need to know the example of secretion system or just basic structure? What do you mean by the example? like each of them is quite different, right? So for each secretion system, you need to know landmarks for each, and you need to have in your head how the picture of secretion system looks. Because I won't ask you to memorize everything, but if you see the picture of secretion system, you need to be able to recognize it. And Rita asks, do we need to know the 3 main carbon fixation pathways details? Yes, you need to know that in details. Specifically that. That's very important. Those 3 pathways for carbon fixation as 4 ways of proton multi-force generation is pretty important for you to know in detail. Umair asks, do we need to memorize gene names? No. I don't memorize gene names, so you don't have to. So if you are struggling to memorize some name of the gene, don't. There are the only genes that I wanted you guys to know are those involved in glutamine synthase regulation, and that's it. Ivan asks, is it Ivan? Yeah. For type 1 secretion system, HLI-beer equivalent to RTXD? Yes, it is, but don't bother memorizing those letters. If you, from this what you wrote, I can definitely say that you understood what I wanted you to understand, so it's all good. Okay. I'm waiting for, okay. If you guys remember something, please write a message or shoot me an email. And now I will respond to carbon labeling, what Rita asked. So when you have glucose is a molecule that has 6 carbons, right? Each of those 6 carbons is marked with a specific number. So you exactly know which one is carbon position 1, which one is carbon position 2, which one is 3, which one is 4, 5, 6. Okay. So each of those 6 atoms of carbon has specific position. So we know what one is 1, what is 2, what is 5, what is 6. Okay. Now, let's say that you want to do that labeling. You want to label glucose with a carbon that is radioactive because you want to see what's happening downstream in the pathway. So that glucose will be metabolized. So at the end, you will get some triiosaphosphates or pyruvate at the end, or acetyl coenzyme A, depending on the pathway, okay? So each of those carbons in glucose, of those 6 carbons, ends up in some specific molecule. Acetyl coenzyme A, pyruvate, carbon dioxide, depending on the pathway. Now, if you label glucose at position 1, so that's the printing, that's the first thing. So if it's on position 1, and let's say this organ, let's say you label it at position 1, and then when metabolism is done, you looked where your radioactive carbon is. So only 1 carbon is labeled. So you have only 1 carbon labeled. And that's carbon position 1. So you label it, and then at the end of metabolism, you see that there is labeled carbon dioxide. So what you can have, if it's aerobic metabolism, you can have acetyl coenzyme A produced from glucose, so pyruvate converted to acetyl coenzyme A, and there will be some carbon dioxide released, okay? So if you see that labeled carbon is in CO2 and acetyl coenzyme A, you don't see, it's not labeled, that tells you that your cells are using EDD pathway. So glucose was labeled at the position 1. Then if cells are running EDD pathway, then glucose will be converted to glucose 6-phosphate, then 6-phosphogluconolactone, blah, 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 all the way to ribose phosphate. and you will see one carbon dioxide going, and that carbon dioxide is labeled because that carbon that ended up in carbon dioxide, if cells are running EDD, that carbon is from position 1. So carbon dioxide molecule of carbon in carbon dioxide originates from carbon 1 in glucose, and you labeled that one. If you labeled carbon at position 3 and cells are running EDD, you won't see labeled carbon. You won't see carbon dioxide labeled. Okay? Something else will be labeled. Does that make sense? But if you labeled glucose at position 1 and you see that acetyl coenzyme A is now labeled, so not carbon dioxide, that means that your cells are going through EMP pathway, because in EMP you will have glucose 6-phosphate, fructose 6-phosphate, then fructose 1,6-bisphosphate, blah, 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 all the way down to acetyl coenzyme A. If it was EDD, you won't have that acetyl coenzyme A, you will have that decarboxylation, and that carbon dioxide will be released. Now, if you label, so this was all when you label glucose at position 1. But you can also label other glucose, other carbons in glucose. You can put that radioactive labeling in position 3. Now, then you will see different mode. If you put label in position 3, then labeled at the end will be carbon dioxide if your cells are running EMP pathway. But if labeled is acetyl coenzyme A at the end, your cells are running EDD pathway. So it's just a matter of which carbon goes in which molecule. That's all what matters. When you think about glucose structurally, if you remember that, let me show you that picture. I think it's I will find you that picture with a molecule that has only C1. Just a second. Let me share my screen and then it will be easier. Can you see this? Okay, so in glucose, you have 6 carbons available for labeling. This is methane, so what you have is only 1 carbon. So things are a little easier to understand. So when you want to conclude whether methanotrophic cells are running EDD or EMP. So this is basic methane oxidation pathway. The red one shows you carbon labeled. Now, if your cells are running EDD pathway, so look this, hexulose 6-phosphate will be converted to fructose 6-phosphate, and track this red carbon. Look this red carbon here. So from formaldehyde, you get hexulose 6-phosphate. So formaldehyde is converted with ribulose 5-phosphate, is condensed with ribulose 5-phosphate. And then this is that 1 carbon. And now what you're doing, you are tracking where this red one is going. So this 6-carbon compound goes to fructose 6-phosphate. So you see here, you see that red carbon, that's what you're looking for. Now, if your cells are going through EDD, this fructose 6-phosphate will be converted to 6-phosphonogluconolactone, and look where this carbon is now. Then you will get conversion to KDPG, and again, this carbon is still here. And then when aldolase breaks this down, you will get pyruvate, because this is where aldolase cuts, and then here, and then labeled carbon goes in pyruvate, the other one goes in glyceraldehyde phosphate. So if you see labeled pyruvate at position 1, see here, pyruvate is here, position 1. That's EDD. Now, what if cells are driving EMP? Now you will have again, this is labeling. You see again, fructose 1,6-phosphate, so still position 1. Now, again, you will have aldolase that will now break this fructose 1,6-phosphate down. And what will you have here? So because it breaks it here, you will have pyruvate labeled at position 3. That's your basic difference. So pretty much you are observing where the carbon from glucose will end up. And there is a specific place where each enzyme cuts. And since EDD and EMP are different pathways, there are different enzymes involved, and they cut in the different positions. So based on that, you will see labeling in different molecules. I hope it's a little more clear now. Oh, I know this is not easy, so don't worry. Any more? Let's see. Questions? Are we okay? I don't see any more questions, so think about, do you have any more questions, or we can log out for the night? Okay, Ryan says, when we come tomorrow, can we just sit? Yes, yes, you can sit wherever. We will have coffee, so. That's it, are we good to go? Thank you, no worries, Melissa, my pleasure to help. And again, thank you all for a great semester. Please do well tomorrow so I can be happy and proud. That would really mean a lot to me. And don't worry, we will be with you all the like all time, and you know that I'm open for questions. So I can't really tell you answers, that would be non-ethical, but I can definitely explain if something is unclear. I will stop recording then.